Hi students, next we are going to discuss about the inhibitors in biochemistry. Actually we have three situations in biochemistry for inhibitors. The number one inhibitor is electron transport chain. What are all the inhibitors in all the four complexes in electron transport chain is number one. Number two inhibitor is glycolysis, TCA cycle and heme synthesis. I am giving 3 plus 1, 7 examples for glycolysis, TCA cycle and heme synthesis. Third one is inhibitors 3 that we will discuss in later. In glycolysis we have 3 inhibitors, that is glycolysis cycle where it will block or where it will affect the enzyme and also affect the pathway, right? First one is fluoride. Fluoride will inhibit enolase, which is the step involved in 2-phosphoglycerate to phosphoenol pyruvate, right? Fluoride is the inhibitor. Enzyme name is enolase, that is dehydration step. In glycolysis, the step is 2-phosphoglycerate is converted into phosphoenol pyruvate. Okay. Second one is iodoacetate. This is not much significant, but you should know the role of iodoacetate, which inhibits glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate dehydrogenase. Next one is arsenite. This is very important. That is arsenic acid, which inhibits again glyceraldehyde 3-phosphate dehydrogenase. Here the step is glyceraldehyde 3-phosphate is converted into 1,3-BPG. BPG means bisphosphoglycerate, right? So arsenite inhibit the enzyme of glyceraldehyde 3-phosphate dehydrogenase, which converts glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. So these three enzymes are inhibitors are very important MCQ from glycolysis. Next one is TCA cycle. Again we have three inhibitors. Number one arsenite, number two fluoroacetate, number three malonate. This arsenite inhibits the enzyme of alpha ketoglutarate dehydrogenase, which involves the conversion of alpha ketoglutarate into succinyl CoA. Right? This is irreversible step in TCA cycle. Second one is fluoroacetate. This inhibits the enzyme aconitase, which converts the citrate into isocitrate. Here the step is irreversible. And the third one is malonate, which is this same structure analog of succinate dehydrogenase. That is why it will affect the pathway, that is step of conversion of succinate into fumarate, right? So malonate inhibit the action of succinate dehydrogenase. And the third one is heme synthesis topic. We have one inhibitor, that is lead, which inhibit the second step of heme synthesis, that is conversion of ALA, alpha levulinic acid, converted into parfobilinogen. Here the enzyme is ALA dehydratase, that is the step is dehydration step. One molecule of water is released. Conversion of ALA to PBG, that is parfobilinogen, right? So these three topics, glycolysis, TCA cycle and heme synthesis, coming under inhibitors 2, right? In this inhibitors 2, the important 2 points are, number one is fluoride. Why we want to add fluoride while the blood collection from the for the estimation of glucose, right? Suppose if you want to estimate the glucose from the patient, you have to take the test tube with this fluoride for the blood glucose estimation. Why? Because this here the enolase cofactor is magnesium. This is also another important MCQ. Cofactor for enolase is magnesium, right? So fluoride inhibit this enolase while collecting blood for estimation irreversibly. What is the importance for this fluoride? Addition of fluoride for the blood glucose estimation in the test tube, which decreases this enzyme enolase. Or otherwise what will happen, if the fluoride is not adding to the container, the available glucose in the blood sample is automatically metabolized by the blood cells. So what will happen? The blood glucose level will be decreased, right? That is why you have to add fluoride in the test tube for the blood glucose estimation. Otherwise, the whole glycolysis pathway will be blocked or stopped. You will get false negative report for the patients, right? This is the significance of enolase and fluoride. Okay. Next one is arsenic. Usually this arsenic acid inhibit the enzyme complexes, 3 enzyme complexes, very very important complex: alpha ketoglutarate dehydrogenase, pyruvate dehydrogenase, and branched chain alpha keto acid dehydrogenase. These 3 complexes, they require for their metabolism lipoic acid as a coenzyme. Got it? Lipoic acid is act as a coenzyme for these 3 complexes of enzymes, that is alpha ketoglutarate dehydrogenase, pyruvate dehydrogenase, and branched chain alpha keto acid dehydrogenase. So this arsenic acid will inhibit this lipoic acid, that leads to inhibit all the 3 pathways or the enzyme complex. Okay. Well, coming to inhibitors 3, we have 3 topics, important topics in molecular biology, that is replication, transcription, and translation. For all the 3 reactions, we are having inhibitors, right? The first one is replication. Here the inhibitors are DNA gyrase, that is topoisomerase 2, topoisomerase 1, DNA polymerase, and thymidylate synthase. These are all the enzymes involved in replication. The inhibitors are either antibacterial drugs or anticancer drugs. Usually molecular biology, these 3 reactions are always affected by antibacterial agents and anticancer agents. You have to mention 1 or 2 examples for all the enzyme inhibitors. Here the topoisomerase 2 inhibitors are ciprofloxacin and nalidixic acid. Second one is topoisomerase 1. The inhibitors are adriamycin and doxorubicin. Third is DNA polymerase. Here the inhibitor is 6-mercaptopurine. Fourth one is for replication thymidylate synthase. The inhibitor name is 5-fluorouracil. The last 2 is anti-cancer agents. First one is antibacterial. So you have to mention the name with the inhibitor. This is for your short notes as well as MCQ questions. Sometimes in your Viva also some examiner will ask about, tell me some inhibitors for replication, tell me some inhibitors for transcription and translation, like that. Usually MGR University, definitely 1 question will be there from this inhibitor, right? Second reaction is transcription. Here I am giving only 2 drugs only. This is enough for you. One is actinomycin D and another one is rifampicin. Actinomycin D is the anti-cancer drug, whereas rifampicin is the anti-tuberculosis drug. This actinomycin interfere with the DNA strands, whereas rifampicin interfere with the inactivate RNA polymerase. The role of rifampicin is it inactivates the RNA polymerase. One more drug inhibitor for transcription is alpha amanitin. Suppose very rarely some examiner will ask in the MCQ question, the name is alpha amanitin. This is also inhibitor of transcription, right? Third one is translation. Here we are having 2 groups, prokaryotes and eukaryotes. Sometimes the exam, in your exam, the question will be explain the translation for prokaryotes, explain the translation for eukaryotes, like that only. That is why I am giving 2 separate titles, prokaryotes inhibitors and eukaryotes inhibitors. Prokaryote inhibitors, all the 3 antibacterial agents, tetracycline, erythromycin, chloramphenicol. Whereas in eukaryotes, diphtheria toxin, puromycin, cycloheximide, right? Where it will affect and how it will inhibit the pathway is very very important. Here the streptomycin, tetracycline, chloramphenicol, erythromycin all are affect the binding of ribosomal units, that is 50S, 60S, 80S, like that. in the ribosomal complex, it will bind the site, that is 50S site, 30S subunit like that, and it will inhibit the initiation or reading like that, elongation like that. Whereas puromycin and cycloheximide, they will affect the amino acid tRNA, that is chain termination, whereas cycloheximide affects the peptidyl transferase, right? Cycloheximide affects the peptidyl transferase enzymatic activity of the 60S ribosomal subunits. Don't worry about the ribosomal subunit names. You just mention about the prokaryotes, tetracycline, erythromycin, chloramphenicol, and eukaryotes, diphtheria toxin, puromycin, and cycloheximide. Okay.